what's in a name? That's what this episode is all about. Taxonomy, how we classify and name animals. And that's no small task, because as we saw in the previous episode, we've already classified around 1.5 million species of animals so far, and we're still discovering more species every day, and we have to come up with new names for each new discovery. Not only are we discovering new species all the time, but we're also refining the ones that we've already classified and named. For example, there may be some animals that we've grouped into a species before, but it turns out that they're actually two separate species because, say, they don't interbreed, for example, or perhaps they have these behaviors that we hadn't noticed before. For example, up until 2016, the giraffe had been classified as one species. But after examining DNA evidence, scientists now recognize 4 species of giraffe, as distinct from one another as a brown bear is from a polar bear. The biological definition of a species is groups of organisms that can interbreed and produce viable offspring. If they can't interbreed, they are a separate species. There are many reasons why organisms couldn't interbreed. For example, it could be for the very simple reason that they don't breed at the same time of the year, so there's no genetic mixing between the two. Over the course of hundreds or even thousands of years, these two populations would become different enough from one another to be considered as separate species. Another reason is that there may be a physical barrier, like a mountain range, that prevents interbreeding, so there's no gene flow between the groups of animals. So again, over time, they would become different enough that we would consider them to be separate species. There are many ways in which we refine our naming system. As I mentioned earlier, we may have realized that something we thought was one species was actually 2 or more species, but it can also go the other way. We may realize that something that we thought was 2 different species are now actually the same. But the point is that we have a naming system for animal species, and this naming system is important for a number of reasons. First and foremost, naming systems are important so that we know what we're talking about when we're talking to one another about these organisms. And secondly, because the names actually mean something and aren't arbitrary jumbles of letters thrown together. We're talking about taxonomy, which is basically the same thing as systematics, and it represents the organism's phylogenetics. And that word biologically implies that there's some kind of relatedness among the organisms that are grouped together compared to those that are grouped apart. And that relatedness will become very important as we see why we named the organisms the way we do, because it's not just meant to be a name, it's meant to be a useful name. A name that can be used in the study of the organisms and of nature in general. Our historical classification system is based on one created in the 18th century by a Swedish botanist named Carolus Linnaeus. He created a hierarchical naming system where the lower you go down the list, the more closely related organisms are. So the higher up you go, the more distantly related organisms are lumped into the same taxonomic grouping. If we were to use humans as an example, we're known as Homo sapiens. The species name has 2 components. It's always a genus plus a species, never just the species, because the genus tells us what group we're in. In the case of humans, Homo is the genus from the Latin for man. There are other species of humans that are a part of that genus, such as Homo erectus and Homo neanderthalensis. The species name tells us which kind of that group we are. In our case, sapiens, the name that Linnaeus gave to himself and the rest of living humanity, from the Latin for wise. We won't get into whether he should have chosen a more appropriate word than wise. But as you can see from this chart, we're nested into higher groups, meaning that there are other kinds of organisms that are closely related to us, but not exactly the same. For example, in our family, Hominidae, there are other great apes, such as orangutans and the chimpanzees. And above that level, we have the order primates. In that higher grouping, there are other types of primates besides just the great apes, including monkeys and tarsiers and lemurs. Those primates are all mammals. And if we lump all the mammals together, we're we're talking about a much bigger variety. Elephants, dolphins, mice, whales, bats. So as we're going higher up, we're getting less distinct. We have more diverse groupings lumped together. This hierarchy allows us to see our heritage with respect to the groupings. As we go further down, it tells us what separates us from the other animals and makes us different. So the way we talk about organisms is to use the two-name system of genus and species, like the example we just saw, Homo sapiens. This is known as binomial nomenclature, which is just a redundant way of saying a two-named name. So why would we use this binomial nomenclature instead of a common name that we're all familiar with? Well, let's say that an international organization like the United Nations is working on a project to increase the honeybee populations. And there are people from many different countries with different languages working on the committee. It quickly becomes apparent that once you're outside of your own linguistic group, it is not useful to use that common name. But by using the taxonomic term Apis mellifera, which is the binomial nomenclature, we all know what it is. Scientific work is international by nature, so that binomial nomenclature is a very practical aspect of doing science. But there's another very important feature of binomial nomenclature, and that is that the names represent something. The names and the groupings represent their relatedness, their relationships with one another over evolutionary time. So that's why these organisms are grouped into what are known as phylogenetic trees. Firstly, because they look not unlike a tree with its branches. And like a tree, it is rooted in the past. The present is at the level of the tips of the leaves. But also because the organisms on the same branches are more closely related to one another than they are to the ones on the other branches. And so, not only do we have names that indicate relationships to one another, but they also indicate the relative closeness or distance of those relationships with one another, which is a factor intentionally embedded in this naming system. As you can see, many of these phylogenetic trees are basically the same. They're sort of a vertical time component rooted in the past, and as you can see at various stages along the way, that there is a branching off process. Some die out, but others persist and evolve. So there is a fluctuating ebb and flow over time. But the naming system represents that too, and takes it into account. Why is the naming system in flux? Well, it's because we're refining the tools we use to understand what the different animals are now that we have molecular techniques and DNA techniques to determine species, whereas in the past, we tried to determine species based largely on superficial characteristics. So, for example, much of what's living in the ocean contains the name fish, because we used to think everything that was shaped like a fish or swam in the water was a fish. But convergent evolution can make completely different species look quite similar, such as these. These are dolphins, a prehistoric ichthyosaur, and a shark. The dolphin is a mammal, the ichthyosaur is a swimming reptile that lived during the time of the dinosaurs, and the shark is a fish, the only actual fish of the three. But they have evolved to look very similar, because they have had to adapt to very similar circumstances. That is known as convergence. The dolphin and the ichthyosaur look like fish. It's because that's the best way to be if you want to swim around in the water and function. They need to be capable of swimming at high speeds to catch prey while evading predators. So they've evolved a body plan that is hydrodynamic, with fins for navigation and a tail that provides them with propulsion. In the past, we might have grouped these organisms together under the same name, erroneously suggesting that they are related to one another, when we clearly now know that they are not. But there's no reason why, just because of superficial similarities, that we would say that they're evolutionarily related to one another. They developed their similarities as a result of convergence to similar needs. Another example would be flight. If we assumed that all these creatures inherited this trait from a common ancestor, we would be wrong. Clearly, these very different animals did not acquire the ability to fly from a common ancestor. Rather, it was their behavior and physiology that evolved side by side. They each had to satisfy some evolutionary requirement of taking to the sky. To conclude, there are many superficial similarities between animals that we can point to. They are products of evolution because we know that's how evolution works, to tailor the form to the required function that the animal needs to perform in order to survive. But those are not good criteria for creating names and grouping systems, because sometimes it would suggest the two organisms should have the same name when they are actually different. Because in reality, they're completely different from an evolutionary perspective, such as the example of whales, jellyfish, and actual fish will attest. The way we do it now is to use truly inherited characteristics and to group organisms based on similarities from inherited characteristics alone. These are known as homologies. Homologies is when organisms have similarities because they're truly the same in the sense that they've inherited them from a common ancestor. Examples of homologies include the human coccyx and the tails of primates, the similarity in cell organelles between plants and animals, placental gestation between land and aquatic mammals. Looking at the forearm of vertebrates now, and turning the page away from the behavior of flight, we see that some, but not all, have evolved flight. When we look at the internal structure of the forearms of vertebrates, we see that they're all made up of the same pieces in the same order. Sure, it'll be in slightly different forms or different arrangements to satisfy different requirements, whether it's to fly, or to swim, or to swing on a branch and grasp things. But from an evolutionary perspective, these forms of vertebrates are all made up of the same bits and pieces, i.e. the same bones in the same relative positions to one another. And that implies that they all got them from the same place, from a common ancestor, and that they diverged after that point to satisfy different requirements. And so, both of these instances of analogous evolution via convergence and homology being inherited are really strong evidence for evolution itself. However, if we want to describe relationships amongst animals, we can't use superficial similarities. We have to use only inherited characteristics that have either remained the same or which have changed over time because of evolution. So now, biologists interested in taxonomy are attempting to restructure our animal family tree to where we're grouping animals together only based on evolutionary relationships and not based on any superficial ones. This requires a very rudimentary understanding of how we group organisms, how we cluster them into these trees. Basically, we'll see that the only true kind of evolutionary grouping is one that involves a common ancestor and all of its descendants. That's what would be called a monophyletic group, which is basically meaning one origin, and so that all of these organisms are from one ancestor. If we look at this imaginary cladogram, organisms 1, 2, 3, 4, 5, and 6 are all related to one another directly via this common ancestor right here. However, there are other grouping systems that were used in the past that sometimes led to strange bedfellows. For example, this group, where we have an ancestor and some descendants, and we say, well, these other descendants, well, let's call them something else. Now that's not a true relationship in the evolutionary sense, because 1 and 2 are part of this group that includes 3, 4, 5, and 6, and to exclude them for one reason or another does not make sense. The family of great apes was formerly known as Pongidae, and humans had their own family. Why? Because, of course, we humans are so special and unique. We wanted to exclude ourselves from that group, but that would be evolutionarily inaccurate, because no matter how special and wonderful we think we are, we and the great apes do have a common ancestor. We have one family name for all of us, and it's now Hominidae for all the great apes, including ourselves. That would be a corrective approach, to change from the previous superficially derived decision to say, well, these other animals seem different enough that I'm putting them in a different category. We can't do that if we're talking about evolutionary relationships. Another very simple example is that birds are essentially dinosaurs. So by creating a class for birds alone, it makes Reptilia paraphyletic, meaning that there's a common ancestor for all the reptiles, and we've taken out one of their descendants and put it in a different group. That's erroneous when you're using taxonomy to describe the evolutionary relationships. A corrective approach would be to put birds as a subset of ancient reptiles, known as archosaurs. That not only includes their cousins, the crocodiles, but also the extinct dinosaurs. The point is that we're trying to build a family tree that accurately represents our evolutionary relationships. And so the names that we are using are important because they're meant to describe those relationships. Not just the names themselves, but also the groupings under certain names, the collective grouping into certain named categories and exclusions into separate categories. And so that's where we're going to go next, is to recognize that building a cladogram, or our animal phylogenetic tree using modern techniques, molecular tools and DNA, for example, that allow us to really measure relatedness and create these groupings. They are meant to be means by which to group organisms that have evolutionary inheritance or heritage in common, and to separate those that are separated by distinct evolutionary events that had created different lineages for those groups. The characteristics that are used to separate and group animals is what we will explore in the next and last part of this episode. We'll answer the question of how do we go about creating these character categories and say, this animal is an arthropod, that animal is not. So they're known as evolved characteristics. Derived traits is another way of speaking about them. And the first one is at the most simple level, that of the cellular versus tissue level. So remember, we go back to the definition of what is an animal. An animal, you will recall from the definition in episode 1, is a multicellular eukaryote that ingests its food. So it's multicellular, but that doesn't mean it needs to have tissues. So the first level of differentiation in our groupings is, do they have tissues or not? If not, they're in one group. If yes, they're in another. And in fact, the most simple animals, like sponges, don't have tissues. They are just a package of cells that happen to work together very well. And so this is the means by which to separate sponges from all the rest of the animals that have evolved tissues. Now, there are 3 kinds of tissues that are formed in embryos. The internal one, the endoderm, the middle layer, known as the mesoderm, and the outer layer, the ectoderm. In higher animals, endoderm gives rise to the gut, and mesoderm to muscles and organs. The ectoderm gives rise to the outer layers of skin. But the thing is that these 3 layers didn't all happen to evolve all at once. In the first simplest animals, they just evolved 2 layers, being endoderm and ectoderm. The third and last layer to evolve is the mesoderm. That's the one that gives rise to muscles and organs, remember. These components of cells versus tissues are the first upon which we start to parse the different animals into different categories, in this first instance being at the sort of level of organization of their body components. So the next level to look at would be, once that there are tissues, what's going on in the embryological development. Now, the embryological development of an organism will always begin with a zygote that divides into becoming a packet of cells. It is then known as a blastula. Up until the development of the blastula stage will be the only embryological stage that is common to all animals. But in the simplest animals, like sponges, the blastula doesn't differentiate or doesn't evolve or develop into anything else. Those cells just remain cells, and that they kind of become specialized and so on, but it really is just a big specialized aggregation of cells. In higher animals, the embryo undergoes further development by beginning to fold upon itself, and this is the origin of the embryological tissue layers. This process is known as gastrulation, which begins with an infolding in the embryo, a process known as invagination that I'll illustrate with the use of this balloon here. So now what we find is that the embryo has both an inner layer and an outer layer that are linked by this invagination. Now this process of invagination that leads to the development of the digestive tract, in simpler animals like jellyfish and polyps, it does not lead to a tube that has an opening on the other end. It really is just essentially a dead end with one hole that acts both as a mouth and an anus to allow inflow of food and an outflow of wastes. In higher animals, that digestive tract will continue to form a tube going all the way through the axis of the animal to create a hole on the other end, therefore making a complete digestive tract that will have an in hole on one end and an out hole on the other. So basically, as a reminder, because it's easy to get off track, we're talking about ways to group certain animals together and to separate others into categories. And the last distinction between the major groups will be once their embryo is developing this complete tube, or has completed gastrulation, what does the blastopore become? Is it the mouth? If so, those animals would be called protostomes, because stoma is the word for mouth and proto means before or first. So it's the mouth first, and it means that the blastopore becomes the mouth of the organism, and as it grows upwards and outwards to the other hole at the other end, it becomes the anus to complete that tube. Somewhere in evolutionary time, there was sort of a polar flip, where other organisms developed in the opposite direction, where the same process is going on, and the blastula folds on itself to create this blastopore as the digestive tract is forming. But here it's the polar opposite, where the first hole that is created becomes the anus. And as that tube grows and creates another hole at the other end, that other hole becomes the mouth. So these animals, which includes ourselves, are known as deuterostomes, because it means mouth second, whereas the first hole is the anus. So from a developmental point of view, it means that at some point in our lives, we were all just anuses. Some of us still are. So that's it. And that's how we can now go back to that animal cladogram and say, well, this is how we get these branches. And note that here is a common ancestor to all animals, being some kind of protozoa within the kingdom Protista. And that the first group of animals to evolve were the ones that developed multicellularity, being the sponges. But they're on their own over here, because they haven't evolved any tissues, unlike the common ancestor of Cnidaria, which are the jellyfish and polyps. They have evolved tissues, however, only 2 layers, so they're known as diploblasts, with endoderm and ectoderm only, which are filled by layers of jelly between them. Further down the animal cladogram, we find the evolution of mesoderm, which means muscles, and all of a sudden this whole group from this point onwards are the true animals with all 3 layers, or known as triploblasts, which have muscles and organs. The next major distinction is between protostome triploblasts and deuterostome ones, as their embryological developments are like polar opposites of one another. Within these 2 principal animal groups, there are many phyla that we will explore together in this series and discover the other evolved traits that distinguish the major animal types from one another. So in closing, every organism has a name that is unique to it and it alone. And the system that this naming is built upon has 2 main features. One, it's hierarchical, which means that the animals are grouped into nested arrangements with other animals that they're closely related to, and they're separated from those that they're more distantly related to. And 2, the words themselves contain meaning, and they give you information about the organism. That's a concept that we're going to explore more in the next episode. Animals are grouped into common categories using shared, inherited, evolved characteristics, which imply that they have a common evolutionary history. Other animals that have evolved differences over evolutionary time are put into separate categories. Since our current estimate of the number of animal species out there is about 1.5 million, and we're constantly reclassifying others as we learn more and more about them, we'll be needing to create a lot of new names for the foreseeable future.